In today's session, we'll be discussing vitamin B12, and the other name of vitamin B12 is cobalamin. And so let's get it. So vitamin B12, let me take pen. Vitamin B12 is required in humans. We require vitamin B12 in two essential enzymatic reactions. One is the reaction which is catalyzed by the methionine synthase, the one which we usually remethylate homocysteine to methionine. I hope you can remember that reaction when we are activating when information of S-adenosine methionine. That reaction is very important. Also in the isomerization of methylmalonyl to form succinyl CoA. Okay, and remember that some sources of methylmalonyl, threonine, methionine. Also, you can get methylmalonyl from propionyl CoA, and propionyl CoA originated from odd chain fatty acids, also from isoleucine and valine. Okay. Then, when cobalamin is deficient, you will have accumulation of branch chain fatty acids, which will become incorporated into the cell membranes, including those of the central nervous system, and that can explain the manifestation of neurologic disorders which are associated with cobalamin deficiency. Okay, note that folic acid, which in form of N-methyl tetrahydrofolate, is required in the remethylation of homocysteine. So if one has deficiency of B12, folate in form of 5-methyl tetrahydrofolate will be elevated. Okay, and also the levels of homocysteine will be elevated in what is known as homocystinuria. Okay, that patient will have increased levels of homocysteine in urine, homocystinuria. Now, here we have those two reactions: conversion of homocysteine to methionine, reaction catalyzed by the methionine synthase. You might find some text calling this enzyme the homocysteine methyl transferase, in which you are transferring a methyl group. You can see here you require tetrahydrofolate in form of N5-methyl tetrahydrofolate. That is why if you have deficiency of B12 here, the N5-methyl tetrahydrofolate will be elevated, and also homocysteine will be elevated, leading to homocystinuria. Also, you can see odd chain fatty acids and some amino acids. The four amino acids are threonine, methionine, isoleucine, and valine. When they undergo degradation, they will give you methylmalonyl CoA. In fact, this should be L-methylmalonyl CoA. Then this methylmalonyl CoA undergoes isomerization, a reaction catalyzed by the methylmalonyl CoA mutase, which requires vitamin B12 in form of deoxyadenosine cobalamin to form succinyl CoA. Succinyl CoA will go to the TCA cycle. It can also be used in either gluconeogenesis or to produce energy. Now, structure of cobalamin and its coenzyme form. Now, cobalamin, let me take a pen. Oh, sorry. Let me take a pen. Now, it contains a choline ring system. Okay, a choline ring system, which resembles that of the porphyrin ring in the hemoglobin. You can remember the porphyrin ring in heme. Also remember your chlorophyll. Chlorophyll also has a porphyrin ring system, but it differs in that the choline ring, two of the pyrrole rings are linked directly. Okay, remember the pyrrole rings in porphyrin are linked via methane bridges. Okay, but in choline ring, two of the pyrrole rings will be linked directly rather than through a methane bridge. Okay, then at the center we have cobalt, and like in heme we have iron, and in chlorophyll we have magnesium. Here it is cobalt at the center of the choline ring. Okay, then it is held by 4 coordination bonds with nitrogens of the pyrrole rings. Then the remaining coordination bonds, one is linked to the nitrogen of the 5,6 dimethylbenzimidazole, and in commercial preparation, the other, remember, nitrogen can bond with six coordination bonds. So the four, not the nitrogen, the cobalt at the center, it can link. So here, if we have cobalt here, it can link with 4 nitrogens. Okay, then one of the bond here will link with a nitrogen of the 5,6 dimethyl benzimidazole. Then the other coordination bond will link with the cyanide, and either link with cyanide, that is in a commercial preparation of the vitamin in form of cyanocobalamin. And in the physiologic coenzyme, this cyanide is replaced with deoxyadenosine to form the 5' deoxyadenosine cobalamin, and also the 5-deoxyadenosine can be replaced with a methyl group here. So this is the position where it is changing, and I will show you that to form. If that is replaced with the methyl, we will form methyl cobalamin, which are the coenzyme forms of the enzyme, okay, of the vitamin. Sorry. As I have said, cyanide can either be replaced with a 5' deoxyadenosine to form 5' deoxyadenosine cobalamin, or a methyl group to form methyl cobalamin. Now here is the structure. You can see these are the pyrrole rings. Okay. These 4 of them, you can see nitrogen. The nitrogens of the pyrrole are linking with cobalt. Then you can see cobalt here. It is linking with this compound here, and you can see this is a benzene ring, and this is an imidazole ring. Okay, and you can see it is at position number 5 here. This is carbon number 5 and number 6. So it is the 5,6. These are methyl group. 5,6 dimethyl is a benzene ring, benz, then midazole. So 5,6 dimethyl benzimidazole. Okay. Then the other coordination bond here can either be linked with cyanide, okay, in the commercial preparation to form cyanocobalamin, but the coenzyme form is either methylcobalamin, here you have a methyl group. Also can link it to this. This is the, you can see this is adenine. So, and this is 5-deoxyadenosine. So when 5-deoxyadenosine replaces cyanide there, it will form 5-deoxyadenosine adenosine cobalamin. Okay, this is the coordinating system. Okay, and this is the structure now of B12. Now, distribution. Where can you get this B12? It is synthesized only by microorganisms. Please note that. Bacteria, anaerobic bacteria. Okay, it is not present in plants. So, vegetarians, they must be given supplements because cannot find it in plants. Animals obtain the vitamin preformed from their intestinal microbiota or by eating foods derived from other animals. Okay, so the common source in your gut, the intestinal microbiota, they can produce this, but not in sufficient form. But so you have to derive it from eating other foods derived from other animals. Now, cobalamin is present in appreciable amounts in the liver, red meat, fish, eggs, dairy products, and nowadays, fortified cereals. I know you're wondering, I've said the plants don't produce B12, and why are the cereals here? They must be fortified. Fortified, when fortifying them, you add also vitamin B12. So, folate trap hypothesis. Now, remember the reaction I told you about methionine synthase, where we are using N5-methyl tetrahydrofolate to methylate homocysteine to form methionine. Now, I want you to put that in mind, then we go through this. Now, the effects of cobalamin deficiency are most pronounced in the rapidly dividing cells, such as the erythropoietic cells and the mucosal cells of the intestine. Now, such tissues, they require both N5 and N10 methionine. this N5N10 methylene tetrahydrofolate is required in the immediates in this reaction where you are converting the deoxyuridine monophosphate to deoxyuridine, not deoxypyrimidine monophosphate. Okay, and also they require the N10 formyl groups of tetrahydrofolate. The N10 formyl are required in the reaction number 3 and reaction number 10 in purine synthesis. The reaction where we were adding a formyl group to glycinamide ribonucleotide, reaction catalyzed by the glycinamide ribonucleotide transformylase, and also the reaction where we are converting the reaction which was being catalyzed by the 5-aminoimidazole 4-carboxamide transformylase. Reaction number 3 and number 10 in purine synthesis. You can look at that. Those two reactions, they require the tetrahydrofolate in N10 form. Okay. However, in a B12 deficiency, the utilization of N5-methyl form of tetrahydrofolate in B12 dependent methylation of homocysteine to methionine catalyzed by the methionines in this. The one I've also seen some text referred to it as the homocysteine methyl transferase is impaired because you don't have B12 now, and because the methylated form, the N5-methyl tetrahydrofolate cannot be converted to the other forms of tetrahydrofolate. Folate is trapped in this form, okay, and then it will accumulate, okay. So the N5-methyl tetrahydrofolate form will be very high, but this will lead to deficiency of the other forms of tetrahydrofolate, the N5N10 methylene tetrahydrofolate and the N10 formyl tetrahydrofolate forms. They will be very low, okay. So thus, during cobalamin deficiency, there will be a deficiency in these forms, and these forms are required in purine, and purine synthesis will require the N10 formyl tetrahydrofolate in reaction number 3, the reaction number 10, okay, the Ga formyl transferase reaction, and the ICA formyl transferase reaction will require the N10 formyl group, and also in the synthesis of limiting monophosphate catalyzed by the immediates in this, you require the N5N10 methylene tetrahydrofolate, and because they are in deficient forms, you cannot synthesize your purines and TMP. So there will be symptoms of megaloblastic anemia, okay, because you cannot form DNA. So the reticulocytes cannot replicate. So their levels in the blood increase, leading to megaloblastic anemia. Now, which are the clinical indicators of cobalamin deficiency? Now, in contrast to other water-soluble vitamins, 2 to 5 milligrams of the vitamin are stored in the liver, especially in the body, in the liver. Okay. As a result, it may take up to even 4 years for clinical symptoms of B12 deficiency to develop as a result of a decreased intake of the vitamin. Okay. As long as you are taking in the vitamin, it will take a very long time for one to have the deficiency symptoms. But deficiency will happen quickly in months if there is malabsorption of the vitamin. Okay. Then we had this test which was being utilized, but it is not used nowadays, known as the chilling test. It had 2 stages. I want you to go and look at those stages, but they were using a radiolabeled B12. Okay, can go and look at that. Then B12 deficiency is usually now determined by the level of methylmalonic acid in blood. So if you have elevated levels of methylmalonic acid in blood, it shows that you cannot convert your methylmalonyl CoA to succinyl CoA, a reaction catalyzed by the methylmalonyl CoA butase, which uses cobalamin, vitamin B12, as a coenzyme. So if you have deficiency of the coenzyme, methylmalonyl CoA will accumulate, leading to methylmalonic acidemia. Okay. Now, pernicious anemia. Vitamin B12 deficiency is most commonly seen in patients who fail to absorb the vitamin from the intestines. Now, B12 is usually released from the food in the acidic environment of the stomach. Okay, so food, say red meat, you have taken red meat which has B12. Now, in the stomach, the B12 will be released from this red meat due to the hydrochloric acid. Then, malabsorption of the cobalamin in elderly is most often due to reduced secretion of gastric acid, chlorohydria. Now, imagine if you don't have this hydrochloric acid, okay, a condition known as chlorohydria. See, you cannot extract this B12 from the sources, okay? But once we extract the B12, it has a very high affinity for a glycoprotein which is produced by the salivary glands known as haptocholine or the R protein. And this haptocholine or the R protein, you can find some text which usually referred to as transcobalamin 1. Transcobalamin 1, please remember that. And the function of the haptocholine is to protect the B12 from the denaturation from the acid. So it binds to haptocholine after being released, so as it cannot be degraded by the acid. Then this haptocholine B12 complex will move to the intestine. Okay, and in the duodenum, B12 is released from the haptocholine, the R protein, by pancreatic enzymes, especially the pancreatic proteases, the three of them, the trypsin, chymotrypsin, and carboxypeptidase, and binds another glycoprotein which is known as intrinsic factor. So intrinsic factor is produced by the cells in the stomach, the parietal cells or the oxygenetic cells, which also produce HCl. Now, then, after binding to intrinsic factor, the cobalamin intrinsic factor complex moves through the intestine and it will bind to its receptor, cubirin, which is found on the mucosal cells in the ileum, especially the terminal ileum. The terminal ileum is where most of the absorption of B12 takes place. Then cobalamin will be then transported into the mucosal cells. So this once the cobalamin intrinsic factor complex binds to the receptor, the cubirin receptor on the surface of the mucosal cells, it will initiate receptor-mediated endocytosis, and that's how cobalamin is taken into the mucosal cells, then subsequently into the general circulation, where it is carried by its binding protein, transcobalamins. Okay, we have transcobalamin 1 and 2. So transcobalamin 2 will transport like 20%. And transcobalamin 2 is the one responsible for cellular delivery. It delivers this now cobalamin. It delivers this transcobalamin 2 will deliver the cobalamin now to the cells for use. Okay, but 80% will be bound to transcobalamin 1, which was also known as the R protein or the haptocholine. And diagram will help you. So you can see here, you have a diet which is rich in B12. Then here in the stomach, the B12 will be extracted from that food due to the acidic environment. Then it has a very high affinity for the R protein or the haptocholine, which was released in the saliva. So this complex, the B12 R protein complex, is important so as to prevent the degradation of the B12 by the acidic environment in the stomach. Then you can see the parietal cells, which are also producing the intrinsic factor. Then once in the duodenum, this complex is degraded so as to free the B12 by the pancreatic proteases. Then once B12 is freed in the duodenum, it will immediately bind to the intrinsic factor. Then in the terminal ileum, we have receptors for intrinsic factor and the B12 complex. Once intrinsic factor B12 complex binds to the receptors, it will initiate receptor-mediated endocytosis. It is taken up by the mucosal cells. Then it is released into the general circulation, where it will be bound to trans cobalamin. Okay. So let us continue. B12 is taken up and stored in the liver, 2 to 5 milligrams, as I have said. Then it is released into the bile and effectively absorbed in the ileum. So even if you are releasing it to the bile, you are reabsorbing it at the same time. Now, severe malabsorption of B12 leads to pernicious anemia. Okay, when you have severe malabsorption of B12, it will lead to pernicious anemia. This disease is most commonly as a result of an autoimmune destruction of the gastric parietal cells, which are responsible for the synthesis of B12 of the intrinsic factor. Remember, if you have a deficiency in the intrinsic factor, you cannot absorb the B12, okay, because intrinsic factor is required. However, patients who have a partial or total gastrectomy, we remove the part of the stomach or all of it, they will become intrinsic factor deficient, okay, because they don't have parietal cells to produce the intrinsic factor, okay. Therefore, it will lead to B12 deficiency. Now, individuals with a cobalamin deficiency are usually anemic, okay, and they show neuropsychiatric symptoms because remember the accumulation of the branched fatty acids in the neuronal tissue. Okay. Now the central nervous system effects are irreversible. Now, however, pernicious anemia requires a lifelong treatment with either you can do a high dose oral B12 or intramuscular injection of cyanocobalamin. I know you are asking yourself, this person does not have intrinsic factor. Then why should we use a high dose of oral B12? Because supplementation works even in the absence of intrinsic factor because just 1% of the B12 uptake is the one which is dependent on intrinsic factor. Okay? The other can be taken up, okay, by intrinsic factor independent diffusion and simply diffuse. Now, folic acid supplementation usually reverse the hematologic abnormalities of B12 deficiency and therefore can mask a cobalamin deficiency. So remember, we said that what can lead to megaloblastic anemia is that most of the tetrahydrofolate will be trapped in the N5-methyl form, but the N10-formyl tetrahydrofolate and N5-N10-methylene tetrahydrofolate forms will be deficient. So if one has folic acid supplementation, this can give us the hematologic abnormalities, okay? So as to prevent the later central nervous system effects of B12 deficiency, which is the therapy for megaloblastic anemia, both B12 and folic acid are used, okay, until the cause of anemia can be determined. So if one has an anemia, you don't know if one has a megaloblastic anemia, it can be either due to B12 deficiency or B9 deficiency, folic acid deficiency. So at first you don't know which is which. So that is why it is advisable for you to initiate the treatment using both B12 and B9 supplementations, okay? Until you know the cause, okay? Because you don't want to use B9 to treat megaloblastic anemia, which will lead, and if you use B9, the megaloblastic anemia will be, there'll be improvement, but if it had been caused by B12 deficiency, the CNS, the neuropsychiatric symptoms will develop, okay? So you want to use both so as to prevent the development of the neuropsychiatric symptoms, okay? So I hope you have understood that, and we have come to the end of that session. Thank you for your attention, and please subscribe for more content. Thank you.